Thank you, Thera. So I just wanted to point out that we heard from the patient perspective, we heard from the clinical specialist perspective, and I'm going to be presenting the researcher perspective. As all of you know, I know the majority of people that are present here today, I have been a true VHL nerd since 2003. I'm a VHL geek, I believe is my official title. When I started my own lab and I chose to focus on VHL. In 2004, I attended my very first one of these meetings in Japan. And it was hilarious because I submitted 6 abstracts and I ended up getting 6 talks. So I went to Japan. I dragged my boss with me because I said I was no longer going to be, there was no way in the world I was going to do 6 talks. So he did 3 of them and I did 3 of them, and we had a wonderful time. But for me, the most eye-opening thing about that meeting in Japan was meeting the patients and hearing perspectives. So as someone who's intrigued by kidney cancer and how we can creatively shift the story of kidney cancer, and that's where I was looking at VHL as being really about kidney cancer. And when I actually met the patients, when I actually talked to them, I realized that that was just a very small part of their concerns. And the world of advocacy really opened to me at that moment. And I realized that I was asking some of the wrong questions as a researcher, and that the only way forward is really through a perfect dovetailed, or in my case, fishtailed combination between the patient perspective and the researcher voice. And so I became active in the VHL patient organization in the Netherlands. We are a fairly small country, so there's only about 400 patients in the Netherlands. And we were quickly able to tie really a very tight community between the clinical scientists, such as Thera, and the patient voices and highly educated patients, wonderful patient groups to work with. We've tried to really make a solid evidence-based, but also advocacy-based program for VHL patients in the Netherlands. And I think that they enjoy some of the best healthcare in the world as a result of that collaboration. So in the spirit of collaboration, it's an enormous privilege to be kicking off this 14th International VHL Symposium. It is such a pity it's not in Amsterdam. It's a beautiful, beautiful city. I've lived here for nearly 30 years. And I love it, love it, love it for so many reasons. And we had taken a lot of effort to make the physical meeting. As you know, I was going to also host it in 2018, but because my health suddenly became very critical at a certain moment, I had to step away from that. And looking forward to 2020, now we're going virtual. Unfortunately, I do hope that there's an opportunity that I'll be attending another one of these meetings physically in the future, because it's a wonderful thing. I do have a few household topics I'd like to touch upon before we introduce our keynote speaker. So I really would like to thank the organizing committee, both on Dutch soil, because we had a wonderful organizing committee here too, and Thera and I and Ronald went through a lot of efforts to produce this wonderful meeting, but also with Eileen and the team at the VHL Alliance office. Fantastic. Thank you for all your help. Without them, we wouldn't be here today. Also, we would not be here today without our sponsors. And with the presenting sponsor, Merck, as being a major effort, we cannot thank them enough, as well as the other sponsors, Ipsen, and also the European Society of Endocrinology, which has granted us an official meeting status and contributed financially to to making this possible as well. Which brings me to accreditation, an also important thing. If you're a clinician and you want to claim your accreditation, we have received accreditation from the European Union. And we will be contacting, there will be an email with the information with regard to that. This meeting is worth 12 points, and that will be transferred to the American colleagues as well. There's no problem with that. If you are in any country, you have questions, please contact me or contact Eileen, and we'll figure it out. But there'll be more information coming through email after the meeting with regard to that. You will need to keep track of the hours that you are on the meeting. That's going to be something we'll need to know, which days and which hours did you attend, because the points are slidable. So if you attended one day, you could receive 4 points, etc. So we look forward to hearing that. There is a there's a poster session, and the poster session is unique here. We encourage everyone to go check out the amazing posters. When you do visit a poster, it would be really great if you could drop a note to the presenting person and let them know that you saw the poster, if there were any questions that were raised, or even just say, thanks for the time and effort. This was an interesting poster. Try and make it as interactive as humanly possible, because otherwise the people will not get the feedback that they would normally receive in a poster session. The posters are open all the time. So during the poster sessions, you can see all the posters. So it's not that some posters are in only some of the sessions. So just do take time and check them all out. There's not that many. So we're very excited about those presentations. We have a very good agenda. We're excited. Thank you all the speakers ahead of time for the effort you've put into doing this. And I think that is the roundup of the various household points that I needed to make. Please let us know if there are technical issues that come up. And we'll enjoy the meeting. It's going to be a fantastic meeting with a lot of interaction, and I just hope you can reach out to people. Use the chat box to ask questions, and not the chat box. Use the Q&A box. It's over on the right side of your screen, up near the top. You'll see something that says Q&A. That's what you push to ask the questions on a given talk. To navigate between the sessions, you will have to go back to the agenda, and then click on the next session that you want to go visit, and then you'll see it. You'll say live stream or live video. Okay, with that, it's a great honor to speak with Bill Kaelin. He's our keynote speaker today. And Bill, you almost need no introduction in this fan. We're filled with lots of VHL geeks here. But you've been in research and VHL research from the very, very start. And it's important to point out that you've been quite exemplary with regard to your closeness to the patient organizations as well. And we think that that's really driven a lot of the questions and relevance for that. Of course, you and Greg Semenza and Peter Ratcliffe were awarded the Nobel Prize last year. Congratulations. I know you've heard it many, many times, but it's brought a lot of attention to the importance and relevance of understanding the fundamentals of rare diseases. And we think that that recognition is so important when it comes to VHL. So, Bill, I give you the word.
Well, thank you, Rachel. Thank you, everybody. I'm sorry this is a virtual meeting, and I just want to also echo what Rachel said. I remember very well when I went to my first VHL Family Alliance meeting in Hawaii back in the 90s, and at that time I was just starting my laboratory. I had been a clinical doctor, but to me, VHL patients only lived in textbooks and in medical board exams. That to actually be able to interact and meet VHL patients and to be inspired by them was just an experience I've never forgotten, and has stayed with me. And I always try to remember why we do what we do. So now the moment of truth. We're going to hit screen share. Okay. And I think they said to do application window. So let's try that. Okay, we're trying that. Okay, and now maybe you can see my slide deck, and maybe we can shift to presenter view. Can you see that okay? Wow, a miracle. That's a miracle. I should quit while I'm ahead. Okay, so here's my disclosure slide. The most relevant one for today is my involvement with Peloton, which was recently acquired by Merck. Peloton developed the HIF2 inhibitors we're going to talk about. Most importantly, here are the many talented young people who've worked in my laboratory on VHL over the years, so you already get a sense I've been doing this for a few years. And I'll try to mention some specific people as I go along. And here's just a partial list of our many talented and generous collaborators. So, of course, I don't need to tell this audience what VHL disease is. I just want to highlight, however, clear cell renal cell carcinoma in red, because hemangioblastomas and pheochromocytomas are interesting, but they're relatively rare, whereas clear cell renal cell carcinoma is the most common form of kidney cancer, and kidney cancer is one of the most common cancers in the developed world. I also highlight it because we've been blessed to have a number of cell lines derived from clear cell renal cell carcinomas, and that's really accelerated some of our work. And so I think we're in desperate need of cell line models, if we can, for hemangioblastomas and pheochromocytomas. Now, we know from the work done largely at the National Cancer Institute in the United States that There's a predictable evolution in the development of kidney cancer in VHL patients. So, of course, to a first approximation, VHL patients have a wild type or normal allele and a defective allele, but initially they're okay because they do have at least one wild type allele. But over time, these patients can develop numerous renal cysts, and the cells lining these cyst cavities, when examined, are actually now effectively VHL null. They've lost or mutated the remaining wild type allele. So at least in the human kidney, VHL loss causes pre-neoplastic renal cyst. And then over years to decades, these patients can develop clear cell renal cell carcinomas. And when you look at these clear cell renal cell carcinomas, of course, they're still VHL defective, but they've now acquired certain stereotypical mutations involving other genes. And these presumably reflect cooperating events that were required in addition to VHL loss to cause the tumor. So from this, we can conclude that inactivation of VHL is not sufficient for renal carcinogenesis. And we can also conclude that, at least in this setting, VHL loss is the initiating event in clear cell renal carcinogenesis. But of course, here the deck was rigged because the person already inherited a defective version of the VHL gene. But frankly, a very similar picture emerges if you look at sporadic clear cell renal cell carcinomas, that is to say, non-hereditary clear cell renal cell carcinomas. This, of course, is the work this is the work of Charlie Swanton and co-workers. What they've done is to do deep sequencing of spatially distinct regions of kidney tumors, including, when possible, of metastatic deposits, and then have used mutant allele frequencies to infer the evolutionary histories of those tumors. And so almost invariably, they again see that VHL loss is the initiating event. And then you can then see these other cooperating mutations that occurred later during tumor evolution. But many of these late events occurred in a subclonal fashion. You can see different branches, if you will, in the evolution of this tumor. So this has given rise to the idea that there are so-called truncal mutations that are shared by all the clones, as well as branch mutations that are private to specific subclones. And I would argue, based on first principles, that maybe some of these late mutations are only tolerated because they arose in the context of a VHL loss. And for that and other reasons, I would argue we should really be focusing our attention from a therapeutic point of view on the vulnerabilities that are created by VHL loss. Now, if you were a naysayer, you might say, well, maybe by the time you're a full-blown kidney cancer, maybe VHL function is no longer relevant. But we've gone on and addressed that over time, and I'll get back to that in a moment. So we learned over time, of course, that the VHL protein is part of the ubiquitin ligase complex that in the presence of oxygen binds directly to the alpha subunit of the HIF transcription factor and targets it for proteasomal degradation, whereas when oxygen levels are low or the VHL protein has been mutated, now HIF alpha can accumulate, dimerize with its partner protein, which is variably called HIF beta or ARNT, and activate various and sundry HIF target genes. So one of the early questions in the field was, okay, by the time you are a full-blown clear cell renal cell carcinoma, does VHL function matter anymore? Would these cells still be sensitive to, for example, restoration of VHL function? And if so, is HIF really the driving force in the development of these tumors? So years ago, Othon Iliopoulos in the lab took VHL null renal cell carcinoma cell lines. He restored the function of the VHL protein by stable transfection, although we later did it with retroviruses. But in either case, these cells could still grow pretty happily on a plastic dish, but they lost the ability to form tumors in nude mice. And then to address the role of HIF, Keiichi Kondo in the lab took these cells and introduced into these cells a version of HIF alpha, and specifically HIF2 alpha, that can't be recognized by the VHL protein because of specific point mutations. And this restored the ability of these cells to form tumors. Conversely, when Keiichi took VHL null renal cell carcinoma cell lines and eliminated HIF2 alpha, and initially he did this with hairpin technology, but we've done it later with CRISPR, he saw that, like restoration of VHL, these cells could grow happily, or pretty happily, on a plastic dish, but they lost the ability to form tumors in nude mice. And I should point out that this was really a specific property of HIF2 alpha. So HIF-1 alpha, the better studied member of the family, doesn't do this. In fact, if anything, it had the opposite effects. And so this really pointed to HIF-2 alpha being sort of the bad guy here. But I want to digress for a moment and talk about these experiments because I think there are a few important points to make. So this, for example, is a typical mouse from one of Keiichi's experiments where he took a VHL null renal carcinoma cell line, and then he infected it with an empty vector or a vector encoding a HIF-2 alpha hairpin. And in the interest of time, I won't show you the data that this hairpin really does downregulate HIF-2 alpha. And as I just told you, in these cells, they lost the ability to form tumors in contrast to the cells that got the empty vector. So this was exciting, but this is a classical so-called down assay where some complicated process hasn't happened or hasn't happened quite as well. And this turns out to be really important in cancer biology and cancer pharmacology because when you do a down assay, especially, you can be misled. And in particular, you can be misled because of so-called off-target effects. And so to sort of describe this schematically, we frequently add to our cancer cells a perturbant. It might be a drug. It might be a interfering RNA. It might be a CRISPR guide. It's been designed against some target, and we measure some phenotype. But if the phenotype is a down assay, such as a loss of tumorigenesis or a loss of cell viability, we should always worry that what we might really be measuring is a so-called off-target effect. That even though our perturbant does modulate the intended target, maybe it does other things, and it's these other things that's really causing this loss of fitness type of phenotype. Now, I think this problem is actually rampant in the cancer pharmacology and cancer biology literature, and a number of our colleagues in biotech and pharma have written about the fact that frequently when we nominate new cancer targets and we write papers that validate these targets, if you will, in preclinical models, that frequently our colleagues tell us they can't reproduce these findings, or they can only reproduce these findings under extremely narrow conditions. That is to say, they're not particularly robust. And so I've tried to be one of the people sounding the alarm and saying, we simply have to do better when we do experiments, especially if those experiments are aimed at nominating new targets in cancer, new therapeutic targets in cancer. So fortunately, there are some solutions to some of these problems. And so for the off-target problem, the classical solution is to do a so-called rescue experiment. So the idea here is if you can discover or engineer a version of the target that's resistant to the perturbant, and that reverses or rescues the phenotype, it gives you much greater confidence that your effect is on target. And Keiichi is and was very careful, so he did such rescue experiments. So I told you he designed hairpins that would inactivate the HIF-2 alpha mRNA and hence decrease HIF-2 alpha protein production. But he also did rescue experiments. And so here what he did was he took advantage of the degeneracy of the genetic code to introduce translationally silent mutations into a HIF-2 alpha cDNA, such that this cDNA would now encode a modified HIF-2 alpha message that would be resistant to the hairpin, but would still encode wild-type HIF-2 alpha protein. And so now he could do his rescue experiments. So here in both tracks, if you will, the tumor cells got the HIF-2 alpha hairpin. But on the right, he co-introduced this rescue vector that encoded this modified HIF-2 alpha mRNA. And you can see this restored the ability of these cells to form tumors as measured here on the y-axis. So we could say this was on target. And as I also mentioned to you, if anything, HIF-1 alpha and those sort of gain of function and loss of function studies had the opposite effect that compared to HIF-2 alpha. So this really said that HIF-2 alpha was an oncoprotein in clear cell renal cell carcinoma, based again on the work of Chuan Chen and others in my lab. Whereas again, somewhat to our surprise, HIF-1 alpha seemed to be acting as a kidney cancer suppressor. Now, we initially got some pushback on this idea, but you can actually see this very clearly, for example, if you go to the Broad Institute DepMap database. So here what I'm showing you is either enrichment or depletion of guides, CRISPR guides against HIF-1 alpha in genome-wide CRISPR screens in cancer cell lines of different lineages. So the lineages are shown here along the x-axis, and each circle is a different cancer cell line. So if we look just at the kidney cancer lines, shown here with these blue dots, you can see that in many of these cases, the HIF-1 alpha guides are actually enriched over time, as though loss of HIF-1 alpha improves the fitness of these cells. And frankly, we also know that some of these cell lines that don't enrich the HIF-1 alpha guides actually have mutations that have inactivated HIF-1 alpha. So at least under standard cell culture conditions, but Tron's mouse experiments suggest this is also true in vivo. HIF-1 alpha acts as a renal cancer suppressor, whereas, as I just told you, HIF-2 alpha seems to promote tumor growth. So what are we going to do about HIF-2 alpha then? Well, fortunately, by the 90s, a number of companies were developing inhibitors of VEGF, which is, of course, a very well-studied HIF target gene. And it was known that kidney cancers produce very high amounts of VEGF, presumably because of deregulation of HIF, including HIF-2. And we argued that if VEGF inhibitors were going to work in any solid tumor, they should work in kidney cancer. And the good news is that turned out to be correct. So we now have at least 7 FDA-approved VEGF inhibitors for the treatment of kidney cancer. But the bad news is that some patients don't respond to these drugs, and even those patients who do will eventually relapse. So we have to do better. So how can we do better? Well, based on first principles, you might argue, let's target HIF itself, and based on what I told you, let's target specifically HIF-2. But the classical dogma for many years was that HIF-2 wasn't druggable, that it didn't have the right kind of nook or cranny to accommodate a small molecule inhibitor. But fortunately, Kevin Gardner and Rick Bruick, who were then at UT Southwestern, ignored that dogma, and they identified a potentially druggable pocket in the so-called Pas-B domain of HIF-2 alpha, shown here. And they then did screens at UT Southwestern and identified chemical scaffolds that could bind to this pocket, and in so doing, induce an allosteric change such that HIF-2 alpha would no longer bind to ARNT, and hence could no longer bind to DNA. And these initial hits were then out-licensed to a company called Peloton Therapeutics. And Peloton did classical medicinal chemistry to make these compounds more drug-like. They improved their potency, their specificity, and their bioavailability. And they were kind enough to share with us a tool compound called PT2399 that was 1 or 2 atoms different from what was then their lead compound. And Chin Cho in the lab had tested this compound in preclinical models of VHL defective kidney cancer, and she showed that it decreased HIF-dependent mRNAs, it decreased proliferation, such as in soft agar assays, and it decreased tumor growth in orthotopic tumor assays. So that was all very exciting and encouraging, but again, the operative word here is decreased, decreased, and decreased. So these, again, are down assays that you might have seen with any nonspecific poison. So again, the question was, is this on target? So just to show you an example of one of these soft agar assays, here Chin has taken a VHL null renal carcinoma cell line and treated it with 0.2 or 2 micromolar of the inhibitor, and you see a decrease in soft agar growth. But again, in fairness, I could do this with Clorox bleach or any old poison. And so the way to know this is on target is to do a rescue experiment. Here, again, we got very lucky because Bruick and Gardner had already identified a point mutation in the HIF-2 alpha Pas-B domain that would prevent the drug from binding to the pocket, but would otherwise leave HIF-2 alpha largely intact. And so what Chin really did was she used CRISPR to generate isogenic cells that were VHL null, but either now expressed HIF-2 alpha or this drug-resistant HIF-2 alpha variant. And if the cells had the drug-resistant HIF-2 alpha variant, you can see now these cells continue to form soft agar colonies, even in the presence of drugs. So we knew this was on target. And in the interest of time, I'll tell you, she was also able to rescue the effects of this compound in orthotopic tumor assays. Now, as you should, Chin actually looked at a number of VHL null kidney cancer cell lines. I'm missing a hyphen, I think, and found that, much to our surprise, some of the cell lines actually were not inhibited by the HIF-2 inhibitor, despite a pharmacodynamic response. And when tested, these cells could actually tolerate genetic ablation of HIF-2 alpha. So this suggested that maybe there would be some heterogeneity, that we had some cell lines that were HIF-2 dependent and some cell lines that were not. And you can see this also in the DepMap database collection. So now we'll go back to the DepMap. I've introduced this technology before, so now I'm looking at enrichment or depletion of HIF-2 alpha guides in these genome-wide CRISPR screens, and here in the blue are the kidney cancer cell lines. So you can see that some of the cell lines, such as the famous 7860 cells, are clearly HIF2 alpha dependent. They deplete the HIF2 alpha guides over time. But a surprising number of them were independent. And so this suggested that there might be some heterogeneity. And that's exactly what was seen when a second generation HIF2 alpha inhibitor, which is now called MK6482, went into clinical trials in patients with advanced kidney cancer. So these are phase 2 data from patients who had advanced kidney cancer. They had, in over 90% of cases, failed a VEGF inhibitor, and in over 70% of the cases had also failed an immune checkpoint inhibitor. These are so-called swimmers plots. So each horizontal bar is a patient and how long they stayed on therapy. For orientation, the dotted line is 1 year. The patients with the black arrows were continuing to do well on therapy at the time of this analysis, and the patients with the yellow star actually had achieved a partial response on this therapy. So the glass half full here was that some of the patients seemed to be benefiting, and based on this drug has now gone into phase 3 trials for patients with advanced kidney cancer, but some of the patients were not responding. So this is the glass half empty. So there's heterogeneity. Now, again, this was somewhat foreshadowed, you might argue, by the cell line data. Now, I think I now have to do a bit of a mea culpa, because we had suggested that maybe in some cases this HIF2 independence was due to a mutation involving the P53 pathway. That was based on a limited number of cell lines, but now that Laura Stransky in my laboratory has looked at even more renal carcinoma cell lines, we can reject that hypothesis. So here what I'm showing you is a panel of cell lines that are either HIF2 alpha dependent or they're HIF2 alpha independent, and Laura has not treated them or has treated them with a DNA damaging agent called etoposide, which in a P53 proficient cell line should induce both P53 and its downstream targets, such as P21. And on examining more cell lines, she found examples of HIF2 dependent lines, such as this line, TUHR42KB, that is HIF2 dependent, but clearly has a mutation somewhere in the P53 pathway. Conversely, here's a HIF2 alpha independent line that, despite being HIF2 alpha independent, does seem to be able to induce P53 and P21. So we think our initial hypothesis here was wrong, that we don't think a P53 response is critical for HIF2 alpha dependence. And just to drive home this point a little bit further, what Laura Stransky went on to do was to take certain HIF2 dependent lines, such as OSRC2. She completely eliminated P53 using CRISPR. And yet, despite now completely lacking P53, these cells are still sensitive to PT2399 in soft agar assays, the quantification of which is just shown over here on the right. So we no longer think that an intact P53 pathway is required for responding to a HIF2 inhibitor. Now, let's go back to this glass half empty. What else might we do for these patients? So I've had a longstanding interest in synthetic lethality. You may be familiar with two genes are synthetic lethal if a mutation in either gene alone is compatible with viability, but mutation in both genes simultaneously leads to cell death. And it was Hartwell and Friend many years ago who suggested that we should be applying this more in cancer. And so if the A gene was our favorite cancer gene, such as the VHL gene, we should find all the other genes that are synthetic lethal with that cancer gene, and then we should target their protein products, because in principle, the inhibitor of the B gene here should only kill the cells that have the sensitizing mutation, the mutation in the A gene. Now, I will also confess that as drawn here, this is kind of digital. You're either dead or you're alive, but in actual practice, you're either more fit or less fit. But even these sorts of quantitative differences could be potentially therapeutically exploited. Now, one possible synthetic lethal we thought related to MET. So it had been shown by a number of groups, including George Vanderwood, that VHL null renal carcinomas are hypersensitive to the MET ligand HGF. And back in the days, and there's also, I think, a substantial literature now on crosstalk between HIF and MET. And back in the days of the dinosaurs, we did an early synthetic lethal screen using relatively crude technology by today's standards, and using at least hairpin technology, it looked like VHL null cells had a greater requirement for MET than did their VHL restored counterparts. Now, this may or may not have been a red herring, frankly, but it did cause me to whisper to my clinical colleague, Tony Choueiri, that maybe he should test cabozantinib in kidney cancer because cabozantinib inhibits both VEGF and MET. And we thought maybe that would be fortuitous. And he drove the clinical development of cabozantinib for kidney cancer. This is showing you, I think, the registration trial data where cabozantinib was compared to everolimus in patients with advanced kidney cancer. And I think cabozantinib has been approved by the FDA for the treatment of kidney cancer, and we'll return to it a little bit later. Now, another approach to finding synthetic lethals would be to do genetic screens, and there have been a number of these genetic screens over the past decade, but unfortunately, many of them have not borne fruit, and I'll go over some of the reasons why that might be the case. So many of these screens, people took cells that were either wild type for the gene of interest or mutant for the gene of interest. They were then infected either with a hairpin library, or more recently, people have used CRISPR guide libraries. And then genomic DNA would be harvested at the early time points and late time points, and the abundance of the hairpins and guides would be monitored by deep sequencing. And what you would be looking for here is for hairpins and guides that got preferentially depleted under the mutant conditions. So that all sounded fine as drawn on the blackboard, but I think there were several problems in hindsight. One is it's just harder to see the loss of something than the gain of something from a statistical point of view. Secondly, even doing two independent infections introduces some noise into these assays. And then finally, even when these cells were designed to be isogenic, they seldom really were if you consider the heterogeneity even within a given cell line. And as a result, as soon as you pick out a particular subclone from a cell line and start to manipulate it, there will be additional passenger mutations to be sure. So the cells really aren't truly isogenic. So the simple adaptation we've come to is to have tumor cells, and we like CRISPR because we think it has fewer off-target effects. So we engineer our tumor cells to express Cas9. We do one infection with our CRISPR guide library under wild type or pseudo wild type conditions, and I'll give you an example in a moment. And then after editing has occurred, we split the cells to continue under the wild type or mutant conditions. And we've done this successfully now for VHL, RB1, and IDH1. And I should also point out that whenever possible, we complement these CRISPR screens with synthetic lethal screens in arrayed format using isogenic cells and chemical compound libraries consisting of chemicals with known targets. And we also especially like doing RNAi screens in isogenic Drosophila cells. And Drosophila cells, it turns out, gives you two advantages. One is if a synthetic lethal scores in both Drosophila cells and human cells, it's almost certainly robust. And secondly, there are many genes in Drosophila that duplicated during evolution, and as a result, you can get false negatives in human screens due to paralog compensation, but you'll see those in Drosophila where there's only a single member of the gene family. So just to give you an example, Hillary Nicholson took VHL null renal carcinoma cells expressing Cas9, but also introduced a DOX-inducible VHL, and she infected them under the cover of DOX, and then later split the cells to continue under DOX or remove the DOX. She did a parallel screen where instead of using a DOX-inducible VHL, she treated the cells with a HIF2 inhibitor. Now, admittedly, VHL does other things than regulate HIF2, but it's certainly a major thing that VHL does. And then she continued or removed the HIF2 inhibitor. One of the true positives from the screen was CDK4 and its paralog CDK6. So the ancestral family member scored in her Drosophila RNAi screen. It scored in her human chemical synthetic lethal screen using a couple chemical CDK4/6 inhibitors. It did score in her human CRISPR screen with the inducible VHL, but only because cyclin D1 scored, because cyclin D1 is the partner for CDK4 and 6. It turned out CDK4 and 6 themselves did not score because, again, of that paralog compensation problem. They're largely redundant. And to our surprise, this turns out to be a HIF-independent synthetic lethality. This is not driven by high levels of HIF. So just to show you what a validation experiment might look like, Hillary took 7860 cells and reintroduced VHL together with a red fluorescent protein, or she just introduced the green fluorescent protein without VHL. She mixed them 1-to-1, and here she's treating them with a CDK4/6 inhibitor, abemaciclib, and you see a nice outgrowth of the VHL plus cells at the expense of the VHL minus cells. These inhibitors were also active in orthotopic tumor assays. And again, I remind you, this synthetic lethality is not HIF dependent. So on the left is a HIF2 dependent line. Here's a HIF2 independent line. In both cases, after 28 days of palbociclib, a CDK4/6 inhibitor, you see a nice prolongation in survival. Now, Hillary also began to model whether you could combine a CDK4/6 inhibitor with a HIF2 inhibitor in these sorts of assays. And so now we'll go back to that competition assay where the VHL proficient cells are red and the VHL defective cells are green. So now if you look at two HIF2 dependent lines, such as 7860 and 8498, now if you add the HIF2 inhibitor, you see even more dramatic enrichment of the VHL proficient cells compared to the VHL defective cells, compared to just adding the CDK4/6 inhibitor alone. Whereas if she did this with the HIF2 independent lines, she still saw the palbociclib effect, but it was neither enhanced nor suppressed by the addition of the HIF2 inhibitor. So at worst, if you combine the two drugs, you get the palbo effect, but if the cell line is HIF2 dependent, you might get benefits from both drugs. And in fact, our early modeling, again, with orthotopic tumor models, appears to support this. So here's a HIF2 dependent line, 7860 cells. We're monitoring orthotopic tumor growth in cells that have been engineered to express luciferase. There's a strong trend that the combination might be more effective than either drug alone. And in fact, when Hillary did necropsies on some of the patients that received the combination, they were actually tumor-free. And so based on this work, a clinical trial of abemaciclib, a CDK4/6 inhibitor in kidney cancer, is just about to begin. And if that looks promising, we hope to combine it with PT2399. Now, in thinking about this, we might be able to learn a little bit from breast cancer. So in breast cancer, the combination of tamoxifen and a CDK4/6 inhibitor is now standard of care for hormone-responsive breast cancer. Tamoxifen inhibits the estrogen receptor, which in breast cancer cells controls the transcription of cyclin D1, and D1, of course, is the partner for CDK4 and 6. So you can see how you might get additive or synergistic effects. At least in the HIF2 dependent lines, leaving synthetic lethality even aside, PT2399 is going to inhibit HIF2 alpha, which in these lines controls cyclin D1. You can see this in this plot down here, where on the left I have two HIF2 dependent lines, and you can see downregulation of cyclin D1 with PT2399. And now you come in with your inhibitor of CDK4 and 6. One last experiment I'll tell you about. This is unpublished work we've done with David Sabatini and Heather Keyes, as well as Laura Stransky in my lab. We've done a CRISPR so-called enhancer suppressor screen, where VHL tumor cells expressing Cas9 were infected with a CRISPR guide library. They were initially untreated, and then they were split and either treated with the HIF2 inhibitor or untreated. And we looked for guides that were either more enriched or less enriched, specifically in PT2399 treated cells. And one of the top hits was CDK6. So the guides against CDK6 were dramatically depleted when you added the HIF2 inhibitor. Again, nominating CDK4 and 6 is maybe a good target to think about in conjunction with targeting HIF2. Now, I will point out, I don't completely understand why CDK6 scores here and why we didn't run into that sort of redundancy problem, but this is something we're actively pursuing. That is to say, redundancy with CDK4. Now, another reason for thinking about CDK4/6 inhibitors in kidney cancer are that there are a number of papers that suggest that CDK4/6 inhibitors might also enhance the ability of the immune system to recognize cancer cells. And of course, we've known for many years that kidney cancers tend to be relatively immunogenic. Now, I have to do, and so I think I'll return to that in a few moments, but I do think there's some evidence that CDK4/6 inhibitors might enhance immunogenicity. Now, I want to tell you about one last synthetic lethal screen. This is one that was done in my lab by Chin Jiang. It's the same format I told you about earlier. She's now done this with multiple VHL null renal carcinoma cell lines. And one of the top hits from her screen was the integrin gene ITGAV. So this encodes the integrin subunit alpha V. Now we're going to do those competition experiments, and you can see to variable amounts that if you introduce a guide against ITGAV2, you see an outgrowth of the VHL defective. You see an outgrowth of the VHL proficient cells at the expense of the VHL defective cells in those same sort of two-color competition experiments. Below are just some blots confirming that these guides really do knock down their intended target. You can also do this with pharmacological inhibitors. So it turns out there's a small molecule called selengitide that will block this particular integrin. And when you treat the mixture of cells with selengitide, you again see an outgrowth of the VHL positive red cells at the expense of the VHL defective green cells. This is a small molecule. There's also a large molecule inhibitor that's been developed by Carl Wittrup. And again, you see the same sort of thing, but now you see an outgrowth of the VHL positive cells at the expense of the VHL defective cells. Now, I started to get ahead of myself a moment ago regarding CDK4/6 and immunotherapy. I have to do another mea culpa here because I became a bit of an immunoskeptic over the years because cancer immunotherapy for so many decades had overpromised and underdelivered, and that had sort of left me feeling like this. But I think that changed with the immune checkpoint inhibitors. So here are data in patients with advanced melanoma treated with the checkpoint inhibitor Nivolumab, which inhibits CTLA-4. And you can see a modest but statistically significant improvement in the patients who got the immune therapy. And what was particularly exciting is that there seemed to be a tail on these curves, that some of these patients appeared to be long-term survivors. Of course, you could have argued, well, melanoma is immunogenic. What about other cancers? But, pretty quickly, another so-called immune checkpoint inhibitor, Nivolumab, which inhibits PD-1, was shown to be active in non-small cell lung cancer patients, and non-small cell lung cancer was not thought to be particularly immunogenic. These are so-called spider plots where each line is an individual patient's tumor volume as it's changed over time. And so again, you can see that many of the patients had nice responses. And you may know that these so-called immune modulatory drugs, these immune checkpoint inhibitors, have clearly demonstrated activity in the treatment of renal cell carcinoma. Here's early data from a trial of Nivolumab compared to everolimus, an mTOR inhibitor, and you could see a modest but statistically significant improvement in the patients who got Nivolumab. So I think just like VEGF inhibitors are here to stay, and hopefully the HIF2 inhibitors will arrive, I think immune checkpoint inhibitors are here to stay also. Now, why are kidney cancers so immunogenic? Well, in the case of melanoma that I alluded to a moment ago, we know melanomas are very immunogenic. And this graph shows the work of Gady Getz and colleagues at the Broad showing the mutational burden in various tumor types. And you can see that melanomas have very high mutational burdens. And it's thought that that leads to the production of various neoantigens that can be recognized by the immune system. So that's thought to be perhaps an explanation for the immunogenicity of melanomas. But if you look at clear cell renal cell carcinomas, they're not particularly conspicuous when it comes to their mutational burden. So what might be happening here? Well, I think a potential clue comes from Richard Childs and co-workers at the National Cancer Institute, who many years ago had treated patients with metastatic clear cell renal cell carcinoma with allogeneic stem cell transplants as a source of immunologically competent cells. And they actually observed in some cases complete or partial responses. And they were further able to show that in one patient who had a complete response, this patient now had T cells that could recognize a peptide derived from an endogenous retrovirus, this HERVE. And they could show that this peptide was expressed in clear cell renal cell carcinomas, but not in normal tissues and other cancers. And subsequently, several groups, including groups led by Kim Rathmel and Ben Vincent, have suggested that maybe endogenous retrovirus expression is correlated with the probability of responding to an immune modulator in clear cell renal cell cancer. Now, Childs and co-workers went on to show that in the case of this one particular endogenous retrovirus, it was actually directly regulated by HIF2, which, as I told you, is deregulated when you lose VHL. So you can begin to understand why VHL defective kidney cancers would express high levels of at least this endogenous retrovirus. And Chin-Chin Jiang in my lab has now identified multiple endogenous retroviruses that are likewise controlled by HIF. So we think this is simply an example of many endogenous retroviruses that might be reactivated by HIF. And we're excited about this because this opens up some opportunities to pharmacologically manipulate the levels of these endogenous retroviruses in hopes of perhaps making immune therapy work better. Now, as we begin to test more and more immunotherapies for kidney cancer, we would like to have immunocompetent mouse models, but up to now we really don't. So I showed you this slide earlier. This of course, is a nude mouse experiment. We need mice that have intact immune systems. And so we and others are trying to develop such models. And I'll just briefly tell you about the work that's been done in my lab by Laura Stransky in collaboration with Laura Schmidt and Marston Linehan at the National Cancer Institute. So we obtained a mouse from Feng Zhang at MIT that expresses Cas9 after a so-called LOX cassette is excised using Cre recombinase. These mice also express green fluorescent protein once the LOX cassette has been excised with Cre recombinase. And modeling after work that Feng Zhang, Tyler Jackson, and others have done, we've now introduced into these mice AAVs, adeno-associated viruses, that express not only Cre recombinase, but guides against various genes that are suspected or known to play roles in kidney carcinogenesis. And this appears to work. So here are some MRI images of mice where the AAV was directly injected into the kidney of the LOX-Cas9 mouse. And you can see over time the development of these tumors. The good news is when examined, these tumors have lost VHL and also have the predicted inactivating mutations in cooperating genes such as PBRM1. That's the good news. The bad news is we have not convinced ourselves that these tumors are actually HIF2 dependent. So this is still a work in progress. Now, part of the reason I've emphasized HIF2 inhibitors, VEGF inhibitors, immune checkpoint inhibitors, et cetera, is we have to get to combinations. So this is a fairly famous picture in medical oncology. This is a poor patient who had multiple subcutaneous nodules of melanomas, and his melanoma had an activating mutation in BRAF. And so he was treated with a drug that inhibits BRAF, and you can see he had this almost Lazarus-like remission. But within months, his tumor cells had become resistant to the therapy. And this has led to some fairly influential people, including Jim Watson, to say things like, given the seemingly almost intrinsic genetic instability of many late-stage cancers, we should not be surprised when key old-timers in cancer genetics doubt being able to truly cure most victims of widespread metastatic cancer. But what Jim and others are forgetting is the lessons of the 70s and 80s in terms of combination chemotherapy. And it goes something like this. This is basically a math problem. So even a small tumor like those subcutaneous nodules I showed you can contain 10 to the 8th, maybe even 10 to the 9th cells. Most cancer patients have more like 10 to the 10 to 10 to the 12 cells. So if you have a really good drug like that RAF inhibitor, but 1 in a million cells are resistant, either genetically or epigenetically, to that drug, then you can't cure the patient. And the chance of cure is approximately 0. But if you can combine multiple drugs that are not cross-resistant with one another, now you can make the math work for you. So if you can combine, for example, 3 non-cross-resistant drugs, and the probability of being resistant to any one of these drugs was 10 to the minus 6, then the chance of a cell being resistant to all 3 is 10 to the minus 18. And we know combination chemotherapy works because it's worked for diseases like drug-resistant TB. So this is what the treatment of TB looked like before the advent of combination chemotherapy. It's worked for AIDS, and I would argue it has worked for the cancers that we do cure medically, such as certain types of lymphoma and testicular cancer, as well as others. So we have to get to combinations. Here's data presented at the recent ESMO meeting by Tony Chiuri, where investigators combined an immune checkpoint inhibitor, the anti-PD-1 antibody Nivolumab, with cabozantinib and compared it to the standard of care, which for this trial was sunitinib, a VEGF inhibitor, and you can see a seeming improvement. And so now I think we have to get to 3 or 4 drugs. I can imagine what those 3 or 4 drugs might look like. Others might have better ideas, but I think the data I just showed you suggests that probably combining a VEGF inhibitor and an immune checkpoint inhibitor is a good idea. And so now I think we have to add other active agents. So in closing, when I showed you this trial, I said it was glass half full, glass half empty. But again, these were heavily pretreated patients. And you might wonder, how would this drug have fared if it had been used more in a frontline setting in patients who hadn't seen prior therapies? Because we know with each round of therapy, you tend to select for more resistant cancer subclones. And we were able to convince Peloton to treat 61 patients with VHL disease with this HIF2 inhibitor. And in particular, these were patients who had measurable kidney tumors who had never been treated medically, and they were in careful surveillance programs in an attempt to delay or prevent the need for multiple surgeries and hence to try to preserve renal function for as long as possible. Many of these patients had other incidental tumors such as hemangioblastomas and pancreatic tumors. These data were presented this year at ASCO by Eric Yonash and colleagues. So of the 61 patients treated, about 87% had measurable tumor shrinkage. 40% had a confirmed or awaiting confirmation partial response. The median progression-free survival has not been reached, and the 12-month progression-free survival is 98.3%. And gratifyingly, responses have been seen in some of the non-renal lesions, including, for example, some of the hemangioblastomas and pancreatic lesions. So this is what the swimmer's plot now looks like. You can see here, and as a year with the dotted line. Most of the patients at the time of this analysis were doing well, as shown here with the black arrows, still on therapy. And the yellows and oranges are patients who actually achieved a RECIST partial response that's either been confirmed or is awaiting independent confirmation. So statistics can be kind of dry, so just to put a human face on this, I think some of you in the audience might know that the VHL patients, even before the data were public, were starting to post on social media their scans and their results. And here's a patient, for example, saying, I never thought I'd see this day because some of their tumors are getting smaller or in some cases have disappeared altogether. And then a young man who I haven't met, but who I hope to meet someday, sent me this blog through a colleague. So we'll see if we can get this to operate.
Hey everybody, it's Justin. I just wanted to give you a quick update. I am in a gondola right now in Taiwan. Over there is Taipei 101. The gondola is actually right by the Taipei Zoo. But I just wanted to give you a quick update and say I'm doing well. I'm enjoying my trip. If it wasn't for the PT2977 drug trial, I would have never been able to come out here and do what I'm doing right now. So I just want to thank Peloton, and I hope Merck will fast-track this drug for VHL treatment. So if you guys are listening, hopefully you guys will put it on the market to help VHL. But yeah, keep watching these videos. I'll be making more, and I'll get better at it, and I have to get the angles right because it kind of looks fat.
I think that's wonderful because at that age, you should be worried about whether you look fat in your video and not worrying about what your CAT scan is going to look like in 3 months. I should point out that the FDA has now granted this drug breakthrough status, so we hope it'll be approved soon for the treatment of VHL patients. And with that, I'll thank you very much for your attention and your patience, and I'm happy to answer questions if we go to questions.
Thank you, Bill. You guys can hear me fine? Great, good. So there should be a room-shattering applause at this point, but of course, in the virtual world, we're just all clapping against the air. But anyway, lovely talk. Thank you so much. There have been very few questions. I would encourage everyone to go to the Q&A tab. But I will start off with the first question, which was posed by Thera. And I think it's really interesting that, the advances in so many ways VHL has opened the doors to understanding metastatic renal cell carcinoma and has even led to new to the treatment guidelines and so forth. VHL has been very leading in this respect. But now we're at the situation where we'd like to go the other way. advanced renal cell carcinoma, and you mentioned like the Checkmate 9ER that was presented at ESMO by Tony Shueri. We're getting ideas about the combinations that are so important. And now we want to feed that back into the VHL clinical care. So using the fantastic fundamental work that you've partly pioneered and in combination with many excellent fundamental scientists in understanding the pathways regulating metastatic renal cell carcinoma, which has led to treatments, how can we, and this is theorist questions, what are the most promising developments to expect for VHL patients from this data of evidence, this body of evidence?
Yeah, great question. So first of all, I'm what we used to call a lumper rather than a splitter. I tend to look for the commonalities and not the differences. So to a first approximation, I think what we learn in VHL-associated kidney cancer teaches us about sporadic kidney cancer and vice versa. So I wouldn't be surprised if the therapies look quite similar, at least with respect to kidney cancer. My conjecture would be, however, that the VHL patients might even be slightly more responsive, and it's just a conjecture, but I think of cancer as being like tumblers on a lock, where you had to accumulate a certain number of random mutations before all the tumblers fell into the right position. You finally mutated the right combination of genes. And so it's not surprising that sporadic cancers have tons and tons of mutations, some of which involve those tumblers, but some of them are so-called passenger mutations that can then contribute to drug resistance. So the VHL patients, because they were born with one of the tumblers effectively being in place or close to being in place, my guess is they're going to be genetically slightly simpler if you look at their tumors. Again, this is just a hypothesis or a conjecture. And also because they're in such careful surveillance programs, I hope they won't ever get to the point of having end-stage metastatic kidney cancer. I think a truism in oncology is drugs work better when you treat patients who have lower burdens of disease and in earlier stages. So I hope this augurs well for the management of kidney cancer arising in the VHL patients. Frankly, I hope if they're in careful surveillance programs, they'll never get to the point where they have far advanced disease. Now, the other sort of pleasant surprise or partial surprise from the VHL trial was, I do think there was some evidence that HIF2 played a role or would play a role in the hemangioblastomas, but we didn't have quite the richness of models that we would have liked to have had, but it's very gratifying to see the responses in the hemangioblastomas. So I think the HIF2 inhibitors will be helpful for the management of the hemangioblastomas. And frankly, on the pheochromocytomas, and I know you know, Rachel, the broader term here, I should be saying paragangliomas, I guess. You know, I think in the paragangliomas, I think there was evidence for and against a role for HIF2 alpha in these tumors. But I think the weight of evidence now is that maybe HIF2 does play a role. And so I'm hoping for other VHL-associated tumors, such as paraganglioma, the HIF2 inhibitors will also be helpful.
It's interesting discussion. It's actually one of the questions also from one of the attendees, from Henry Timmers in Nijmegen in the Netherlands. He is asking if you expect that the HIF2 inhibitors would work particularly in metastatic pheochromocytomas, given the pathways you've described.
Yeah. So again, I wish we had better models for human paraganglioma. So then we could do those sort of necessity and sufficiency experiments to ask, is HIF2 or VHL loss only important in the early stages of tumor genesis, or is there an ongoing requirement or sensitivity related to VHL and HIF, even in the metastatic setting? So if I had to bet a nickel, I'm feeling a bit optimistic today, so I'll bet that there will be a dependency on HIF2, but I just wish we had better models to answer that very important question, because there are examples of tumors where the initiating genetic events are important early on, but by the time you're a full-blown metastatic cancer, that dependency seems to have been lost. There are even examples of so-called hit-and-run mutations in cancer where there's an effect, but again, the horse is out of the barn afterwards and there's no longer a dependency. So I wish we had better models to answer that very important question. Frankly, we might get the answer from human clinical trials before we get the answer from the laboratory, precisely because we don't have better animal models for some of these tumor types.
Modeling VHL, we have a special session for it, but it's always remained a threshold. The next question is from Professor Ian Frew in Freiburg in Germany, and he has a question regarding CRISPR-Cas screening. Can you comment on the likelihood that you might miss important hits in the case of genes where the copy number is more than diploid, and the probability of achieving a full knockout of these genes decreases with increased copy number? It is likely that tumor cells select for increased copy number of genes that drive the tumor.
Yeah, it's a great question, and there are multiple levels to it. I'm not going to be able to answer all the levels, but what Ian's pointing to is, like everything else we do in the laboratory, there are reasons for false positives and false negatives with CRISPR screens. Just as an example, early on, it was found that if you had a CRISPR guide against a multi-copy gene, whether that gene did or did not play a role in the pathogenesis of that tumor type, you tended to see loss of those CRISPR guides because you were inducing a p53 response by inducing so many double-strand breaks, so effectively a false positive. So I think I take some comfort from the fact that in the current versions of these databases, in some cases there are 10 different CRISPR guides against the same gene. In some cases you can complement those databases with analogous short hairpin RNA screens, because they both have their strengths and weaknesses. But Ian's absolutely right. There are false positives and false negatives. Certainly, if you look at the HIF2 data, that is borne out that the cell lines that the CRISPR data said were HIF2 independent are likewise insensitive to the pharmacological inhibitor and vice versa. So at least with HIF2, that breeds true. You can almost genotype the cells with respect to VHL status if you look at the VHL and HIF guides, et cetera, et cetera. So I think there's a lot of important information there, but Ian's right. There are false positives and false negatives. And then finally, and I think Ian is hinting at this, the other problem with CRISPR is, complete genetic ablation doesn't perfectly phenocopy what a drug's going to do, because a drug often targets one function of what might be a multifunctional protein, and often only causes partial inhibition. So another reason why, like everything else in science, we have to have a little grain of salt.
Right. Okay, so there's a couple more questions. What about hypoxia-inducible factor 3 alpha? Is there more on the horizon? And this is asked by Freda Donskov.
Yeah, for whatever reason, rightly or wrongly, HIF3 has become a bit understudied. So I think we know that it exists in multiple splice variants. We know that many of those splice variants are even potentially dominant negatives with respect to full-length HIF1 and full-length HIF2. But it's really understudied, so I can't say anything intelligent about his question. I think it could turn out to be really important. We just don't know enough.
Okay, thank you. And from MD Anderson, Stephen Wagaspak, thank you for an excellent talk. You mentioned that LOH in the VHL gene is not sufficient for renal cell carcinoma development. What do we know about this in other tumor types, such as PNETs and PHIAs, both of which can also become metastatic?
Yeah, so great question. So first of all, I can even make a slightly stronger statement that even biallelic inactivation of VHL is not sufficient for renal carcinogenesis. I don't know about PNETs and PHIOs, frankly. I'll cheat a little bit and tell you that in hemangioblastomas, I know there are a few other non-random chromosomal changes that people think represent cooperating events. I bet other people on the call know much more about the genetics of PNETs and PHIOs. I would be surprised if VHL loss was sufficient. I assume they're cooperating genetic events, but I'm embarrassed to say I don't know what they are.
Right. And I think there is one request for you to repeat what the combination was. Let me see if I can find it here. Alejandra Cadena asked if you could repeat what the combination treatment was recommended. I think she may be referring to the...
Okay, so let me first of all preface my remarks by saying I no longer have an active medical license, and I don't mean to be giving formal medical advice. So I want to emphasize that in bold, underline, print. I'm not trying to give people formal medical advice. I think the trend right now in kidney cancer care is going to be moving in the direction of either a VEGF inhibitor plus an immune checkpoint inhibitor becoming the standard of care. And you have several choices for your VEGF inhibitor, and you have several choices for your checkpoint inhibitor. There is a camp that thinks that maybe some patients should get 2 immune checkpoint inhibitors, one being anti-CTLA-4 and one being anti-PD-1 or anti-PD-L1, because they think there may be a subset of patients who might even be cured with that combination, even though it's a fairly toxic combination. I can't weigh in there on those two world views. They're not completely mutually exclusive. And the other part of the question was, what do I think could be the third or fourth drug in? And I offered a couple drugs that I think might be useful that we hope to test. I could imagine CDK4/6 inhibitors maybe being the third drug. I can imagine a HIF2 inhibitor being the third drug, or maybe replacing the VEGF inhibitor. I think there's soft evidence for MET. I think the problem with cabozantinib is, although it does inhibit MET, I can't formally prove that has anything to do with its clinical activity. Maybe it's just a wonderful VEGF inhibitor, and that's why it looks slightly better than sunitinib. So I want to be very careful here. I don't know yet the contribution of MET. And other people on the call might have their favorite target, but I do think we have to get to 3 or 4 drugs. Now, I mentioned non-cross resistance. I think that's very important. So there's sometimes a trade-off to be made. So I could imagine combining a HIF2 inhibitor with a VEGF inhibitor being synergistic, because you're blocking VEGF production and blocking VEGF action. But because they're no longer biologically independent, I could also imagine ways you would become cross-resistant simultaneously to both the HIF2 inhibitor and the VEGF inhibitor, at least with respect to angiogenesis. And so that's the trade-off we sometimes have to weigh.
Right. One final question requested by Mark Lodge. It's from the patient perspective. And the question is, can you see a role for the MK6482 or other HIF2 inhibitors in addressing pre-tumor stage cysts?
That's a terrific question. So first of all, I should point out, and I've also declared my financial conflict of interest. I think that's important. Secondly, although the HIF2 inhibitor is very well tolerated at least compared to a lot of classical cancer drugs, it's still not without side effects. And that's very important, because the risk you might be willing to take if you had advanced disease is going to be very different than the risk you would accept if you were feeling fine and just had a few incidental abnormalities on your MRI or CAT scan. So again, the drug is very well tolerated, but patients do develop anemia frequently because you're blocking erythropoietin production, EPO, of course, being a well-studied HIF2 target. That doesn't bother me too much, because you can just give people erythropoietin. The more worrisome side effect is some patients have had a lowering of their oxygen levels in their blood. Usually that's been asymptomatic. It probably has to do with the role of HIF2, both in the carotid body that controls your ventilation, but also, maybe more importantly, in the lung vasculature. So this is probably a concern if people have underlying lung conditions or they live at high altitude. This would need to be monitored carefully. And so I tell you this because, again, if I had to bet a nickel again, I would say that if I was on a HIF2 inhibitor, it would prevent a lot of the manifestations of VHL disease. That's a hunch. That's not scientifically proven. But, some patients might say, but I'm not willing to accept any side effects under those conditions because I feel perfectly fine right now.
Yeah, that's an excellent answer. True. Well, I guess the evidence will tell. I just have one final comment before we end the session. It's a question about, there's so much questions about immunogenicity in renal cell carcinoma. Has anyone tried to use to inhibit BUB-R or something like that to generate more neoantigens to see that there's going to be respect? Is that a laboratory evidence? Is there in vitro evidence for that?
Yeah, it's a great question. Now, as a therapeutic strategy, I wouldn't be that excited because,
but it's just simply
but even if there was a way to do it, of course, if it was subclonal, where each mutation was private or privileged to specific subclones. I don't think that's going to be helpful. But and if the question relates more to could we make a more faithful model of kidney cancer and could we rig the deck by inducing more neoantigens, I think that's a good question. At the same time, as I said, my current bias, and it's just a bias, is that the action could be more at the level of endogenous retroviruses. And frankly, even for melanoma, it hasn't been formally proven that the mutational burden is the key. It could even turn out to be something like endogenous retroviruses.
Exactly. Okay, well, thank you very much. I think we've reached our time limit. So I really appreciate your fantastic talk and the great questions from the audience.